ATP is the primary energy molecule used in living things. The overall function of ATP is to store and eventually release energy. This is done through the ADP and ATP cycle. ATP stores energy in its chemical bonds between the phosphate groups. Since all the phosphate groups are negatively charged, energy is required by the chemical bonds to hold them together. Energy is released when ATP bonds to a protein and the third phosphate group is removed or that chemical bond holding it on is broken. Either sunlight or food consumed by an organism can be used and taken in by the mitochondria to again convert ADP back to ATP, where energy can be stored again. The overall reaction of cellular respiration includes two products and two reactants. The two reactants are glucose and oxygen. Glucose is made during photosynthesis and used during glycolysis. Oxygen is produced during photosynthesis and used during the electron transport chain. The two products are carbon dioxide and water. Carbon dioxide is made during the Krebs cycle as a waste product, and water is made during the electron transport chain and used during photosynthesis. Cellular respiration includes 3 steps. The first step is glycolysis. Glycolysis occurs in the cytoplasm. It produces 2 ATP and 2 NADH that will be used by the electron transport chain. It also produces 2 pyruvic acid molecules that will be used by the Krebs cycle. The second step of cellular respiration is the Krebs cycle. The Krebs cycle occurs in the matrix of the mitochondria and produces 1 ATP, 4 NADH, and 1 FADH2 that are all high-energy electrons that will be used by the electron transport chain. NADH and FADH2 are electron carriers that are helped by hydrogen to carry the electrons. The Krebs cycle also releases carbon dioxide as a waste product. The third and final step of cellular respiration is the electron transport chain, which takes place in the inner membrane of the mitochondria. The reactants of the electron transport chain are NADH and FADH2, which were made during glycolysis. The NADH and FADH2 deliver electrons that travel down a chain of proteins. These electrons are then used to make H2O, which is released as a waste product. The movement of the electrons pumps hydrogen across the inner membrane. The hydrogen then passes through an enzyme called ATP synthase. 24 ATPs are made here and will be used in the cell. Fermentation is a process that converts the pyruvic acid into a different molecule in order to regenerate NAD+. There are 2 types of fermentation, the first being alcoholic fermentation, which is where pyruvic acid is turned into alcohol and carbon dioxide. The second type is lactic acid fermentation. This is when pyruvic acid is turned into lactic acid.